glowing flies and CRISPR-Cas9? How gene editing can help us understand how cancer affects our genome. So what is CRISPR-Cas9 and how does it apply to cancer research? Well, this is Norman. He is a researcher in the Taipale group at the University of Helsinki. He's been setting up a fly lab for functional genomic studies. I asked him to explain what CRISPRs are and how he applies it to his research.
This is basically like the parts of the immune system of of bacteria. So basically how the bacteria defends them against viruses. And then the nice thing is that these CRISPRs basically recognizes DNA sequences. So it's basically RNA based that can bind to the DNA, make complexes and then cut.
CRISPR-Cas9 mechanisms can recognize DNA targets and cut foreign pathogen DNA, but Norman can use it to precisely edit any sequence in the genome. So how does Norman use this for his own research, and how can it be applied to cancer therapies?
Like we are studying MYC, which is one of the major oncogenes. So it's like there's like 5-6 genes like MYC, P10, KRAS, EGFR. Like they are almost in every cancer RB. Almost every cancer cells has a mutation of either of these, because these are the things that drive growth. And so like MYC is one of the most important cancer drivers, and MYC is a transcription factor that activates thousands of genes. It would be like a very good target for cancer therapy. So if you make a drug for MYC, the problem is MYC has no structural features. So one thing that would be equally good as developing a drug of MYC would be choose some of the 1,000 target genes that it activates that is absolutely critical for this growth driving process. So and then develop a drug against this target. So and we tried to identify one of these targets. One of the problems with this is that these targets itself, most of them are essential genes because they're required for driving growth. So now the CRISPRs enable us basically not to delete the gene. It allows us to delete the point in the non-coding DNA where MYC binds to these genes. So and then this allows us now basically, so the gene is basically from the cell used in its normal way, only the input of MYC is lost.
Norman's research uses the common fruit fly as his model organism. It is one of the most studied organisms in biological and genetic research. When screening for cancer targets, why do we start with flies?
So why we use flies? I mean this MYC transcription factor it's super conserved, and because it's like the gene network that drives growth. And now pretty much every multicellular organism needs to grow somehow. So you can find the MYC network even like in some kind of slime molds. So they have almost the same genes with MYC activating these, and this converts from flies, and you can find it in fish, and you can find it in mice, and of course in humans. So the genes are very identical. And so what we try to do is now, because there are so many target genes, we have to do it in larger scale. And I mean, you could of course, in theory, do the same thing with mice. It would be just the cost would be prohibitive. So this is very good to do first this large screening into the flies, and if we find something that is really like a promising candidate, then go up to higher systems. So no, it's like maybe checking in from patient-derived tumor cells whether this has some effect. But I mean, for the initial screening to figure out what is going on, the flies is perfect.
Today, fluorescent markers can be introduced so that they can be easily visualized under a microscope. This aids researchers such as Norman when screening for various target genes.
Bring with the new sequence that you bring in there, or basically when you want to make knockout, bring also like a tag, like a fluorescent protein from jellyfish, or some corals into the flies. And then you just can use with a fluorescent microscope, you can just look at the fly, and then you can select them, so you don't have to use fancy machines and enzymes and sequencing for doing this. Because when you want to do experiments with this afterwards, you always know where your mutation is. So when you have like an unmarked mutation, you have to always do some molecular screening.
Thanks for watching. To find out more, please visit our website and check out our other blogs from the Finnish Centre of Excellence in Tumor Genetics Research.